Hello and welcome dear students. Today we are going to discuss about the topic role of genetic counseling in disease prevention. The main objectives of this module are to understand the concept of heredity and genetics, to learn and understand how genes are inherited, to evaluate the role of genetics in health and diseases, to learn about genetic disorders and their types, and finally to comprehend genetic counseling and its significance. Dear students, let's first have an overview of genetics and heredity. Dear students, before we discuss genetic counseling, let's recap what we mean by genetics and how we can differentiate between genetics and heredity. In colloquial language, we often use the terms genetics and heredity almost interchangeably. However, there is a subtle difference between these two concepts. Heredity is the process of passing genetic information, traits from one organism to its offspring through genes, and genetics on the other hand is the study of heredity, including the variation in inherited traits. In other words, genetics is the study of processes of transferring genes from parents to their children or offsprings. For example, a person's physical appearance such as their skin tone, hair color and texture, eye color, height, etc. is determined by the genetic material they inherit. Apart from these, other characteristics like mental abilities, talents, being prone to specific diseases, etc. can also be affected by heredity. The study of heredity tries to explain how certain traits or genetic material, what we call genes, are passed from parents to offsprings and how they vary from each other. The genetic information is stored in the form of chromosomes, which are present in every cell of the body. DNA is wound up on histone proteins, giving form to chromosomes. Chromosomes are thread-like structures that gradually become visible in the nucleus just before an eukaryotic cell divides. What we mean by visible here is that they are easily seen under a microscope because they take up pigmented stains specifically to stain them intensely. Before the functions of chromosomes were known, they were given this name because chromo means color and somes means bodies. The total number of chromosomes is the distinguishing characteristic of species. For example, in a typical human cell, there are 46 chromosomes present. Dogs have 78 chromosomes, but fruit flies have only 8 chromosomes. The complete set of chromosomes present in an organism is referred to as its ploidy. Each set of chromosomes contains a copy from paternal and maternal cells. Our DNA is densely packed as these sets of chromosomes inside the nucleus are transferred from parents to their offsprings. Each gene is one unit of inheritance. They are segments of DNA molecule, that's regions on chromosomes that code for a specific protein that have a particular physical phenotype. In other words, DNA is a molecule of inheritance made up of sequences of nucleotides that instruct cells to build one or more functional products or molecules which later determine the traits or characters. DNA is the foundational block of life or the molecule of life, which is shaped like a twisted ladder called a double helix. It was discovered by scientists James Watson and Francis Crick in 1953 and gave rise to molecular biology, which ultimately revolutionized the biological sciences. The two later rails of this double helical structure are termed as backbones, and the steps are pairs of the four well-known bases: adenine, thymine, guanine, cytosine, that is A, T, G, and C. In RNA, the thymine base is replaced by uracil. These nucleotides typically consist of a sugar molecule, that is DNA, and ribose in RNA, which is attached to a phosphate group and a nitrogen containing base. DNA and RNA molecules are essentially polymers of long chain of nucleotides. The sequence of these bases constitutes the genetic code that specifies the order and number of amino acids in a protein. In humans, there are about 3.2 billion base pairs that make up the whole genetic material. Scientists estimate that there are over 20,000 genes present in humans, which was discovered after the Human Genome Project results were out. This complete set of genes and other proteins that control the gene expression is termed as the genome of an organism. The human genome is mostly the same in all people with just small variations. Dear students, let's now talk about how genes are expressed and inherited. The expression of genes is the foundation of cell development and differentiation. Genes encode proteins, and proteins in turn direct cell structure and function. The central dogma of molecular biology is the well-established explanation of how genetic information transfers from one cell to another. It states that genetic information flows from DNA to RNA to proteins by processes like DNA replication, transcription, and translation. Although the genetic machinery carries out its expression and division meticulously, sometimes there are deviations in these processes that give rise to genetic anomalies or aberrations or errors. These abnormalities could be in the DNA sequences or chromosomal structures or numbers that are responsible for disease state of humans. The significance of heredity and genetic counseling is going to be our topic of interest in this lecture. It is already recognized that genes are units of inheritance passed down from parent to the children or offsprings containing instructions for making proteins. These genes are present in 23 pairs of chromosomes in every cell of the human body. The two chromosomes in each pair contain the same genes, but they may have different versions of those genes because we inherit one chromosome in each pair from our mother and other from our father. Reproductive cells like eggs and sperms randomly receive one chromosome from each of the 23 sets instead of both, so that a fertilized egg will contain the 23 pairs needed for typical development. Dear students, one important point to note here is that since genes come in pairs, that means there could be two different forms of the same gene. For example, a pair of genes may code for eye color, one may code for blue and the other for brown. These different versions of the same category of genes are known as alleles, where one is inherited from the father and the other is inherited from the mother. Alleles are not the same as mutations that occur in our genome. Alleles are rather small differences in the nucleotide sequences of the same genes within an organism or between two organisms. Mutations are broader in the way we can define them. They are essentially DNA copying errors that occur due to various known and unknown factors anywhere across the genome. Dear students, now moving ahead, let's now understand how do genes affect our health and well-being. Certain variations or mutations or changes in genes can prevent the genes from carrying out its normal function effectively, or at times even halt their regular activity. In other words, some differences in the nucleotide sequence of DNA can lead to incorrectly formed proteins that cannot perform their function, or overperform their function without any regulation, as we witness in cancer cells. In addition, these genetic mutations or variations can impact how a person responds to certain medicines and also increases the susceptibility of a person in the development of a disease or chronic abnormality. Since parents pass their genetic codes on to their children, some disorders tend to cluster in families, just like other inherited traits are peculiar in a family. Dear students, genetic anomalies or abnormal traits or characters that are inherited in a family may not always have an ill effect on our health or well-being. For example, the trait might lead to a nose that is longer than normal or a white patch of skin, but they can also have a slightly unhealthy effect, such as color blindness or difficulty impairment in breathing, as well as majorly unhealthy effect on the quality of a person's life, such as cancer, muscular dystrophy, etc. Some mutations between individuals arise from epigenetic differences, that is, differences in the gene function that are not the consequence of the change in the DNA sequence and may or may not be inherited. With the advent of high throughput technologies, we can now get a picture of an individual's complete DNA sequence and discern genetic abnormalities. These advancements have paved the way for precision diagnosis and medicine that work on therapies and medications that could lead to optimal treatment according to an individual's genetic makeup. Dear students, let's now discuss the genetic disorders and their significance. As we know, human cells have 46 chromosomes. 22 pairs of these chromosomes are autosomes. The 23rd pair of the chromosome is what determines the biological sex of a person. These two chromosomes are called sex chromosomes and are denoted by X and Y. Females are chromosomally represented as 46XX, while males are chromosomally represented as 46XY. That means they have 22 pairs of autosomes and one pair of X chromosome, that is XX in females, and one pair of chromosomes in males, that's XY. There are two replicas of each gene in every pair of autosomal chromosomes, one from each parent. If one of these genes is defective, the other assumes the responsibility of producing enough protein such that no disease conditions develop. In this scenario, the abnormal gene is called the recessive gene. Only if two copies of the abnormal gene are present, the disease may develop. However, if presence of only one defective gene creates a diseased condition, regardless of having a healthy copy of the gene in another chromosome, it leads to a dominant hereditary disorder. In this case of a dominant disorder, one abnormal gene inherited from the either parent is enough for the disease to show up in the child. We refer to the individual with one abnormal gene as heterozygous for that gene, and an individual who received an abnormal recessive disease gene from both parents as homozygous or compound heterozygous for that particular gene. The manifestation of a gene, that's its perceived effect in the form of a disorder, is also referred to as the phenotype in the context of genetic disease. Individuals with a single copy of a recessive disease gene are known as carriers, as they carry the diseased phenotype but do not manifest in their traits. Dear students, let's now focus on the types of genetic disorders. At a fundamental level, nearly all diseases have a genetic component, but how relevant that component is to the disease state and its treatment varies from disorder to disorder. disorders in humans in which genes play a significant role can be broadly classified as single gene defects, chromosomal disorders, and multifactorial genetic diseases. Let's first discuss the single gene disorders. They are also known as Mendelian disorders. They are caused due to a fault in one particular gene. Single gene disorders are characterized by how they are inherited in families. The basic patterns of single gene inheritance are autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. Let's first discuss the autosomal dominant. In an autosomal dominant pattern of inheritance, the anomaly or a group of anomalies typically appear in each generation. Every time an affected parent, father or mother bears a child, the child has about a 50% chance of inheriting the diseased condition. Examples of autosomal dominant are familial hypercholesteremia and Marfan syndrome. Let's now discuss the second one, autosomal recessive. In autosomal recessive inheritance, the parents of an affected individual, commonly referred to as proband, may not have been symptoms of the disease, but they may be carrying the defective phenotype. With each pregnancy, the chance that parents who are carriers can bear children with a genetic disorders is 25%. Male and female children are equally like to be affected. For a child to possess an autosomal recessive disorder, they must receive the abnormal gene copy from both the parents, that is mother and father. Since most recessive disorders are rare, if a couple is in a consanguineous marriage that are related to each other biologically, their child is at increased risk of an autosomal recessive disease or disorder. This is speculated because related individuals have a high probability of inheriting the same rare gene by having a common ancestor. The examples of autosomal recessive disorders are cystic fibrosis and sickle cell anemia. Dear students, the third type is X-linked recessive. In X-linked recessive inheritance, the likelihood of disease getting passed down is significantly higher in males than in females. This is because females possess two copies of X chromosomes and hence they have a chance of having a healthy copy of the gene in the other chromosome, whereas males only carry one X chromosome. Since the defective gene is carried on the X, that is female chromosome, affected males cannot transfer it to their sons as they will receive only the Y chromosome from their fathers. However, in the case of an affected male having a daughter, the diseased gene can be transferred. In females, the presence of one healthy X chromosome covers the effects of the X chromosome with the abnormal one. So, almost all of the daughters of an affected man appear normal, but they are all carriers of the abnormal gene. Each time these daughters bear a son, there is a 50% chance the son will receive the abnormal gene. Examples of this disease or pattern of inheritance are Duchenne's muscular dystrophy and hemophilia A. The fourth condition is X-linked dominant inheritance. In X-linked dominant inheritance, as the name suggests, the abnormal gene manifests in the diseased condition is in females, even if there is also a normal X chromosome present under its dominant character. Males pass the Y chromosome to their sons, therefore, affected males will not have affected sons, but all of their daughters will be affected. In contrast, both daughters or sons of affected females will have a 50% chance of getting the disease. The examples of this type of disorder or pattern of inheritance are hypophosphatemic rickets or vitamin D resistant rickets. Dear students, let's now discuss about the another type of disorders, that is the chromosomal disorders. In these disorders, the defect is due to either an excess or lack of genes contained in a whole chromosome or chromosome segment. For example, Down syndrome and Turner syndrome. Down syndrome is the trisomy of 21st chromosome, whereas Turner syndrome is the condition in which one X chromosome is lacking or missing. Dear students, the third one is the multifactorial disorders. These are caused by abnormal interactions of several genes and environmental factors like drug interactions, medical conditions of the mother during pregnancy, toxic chemicals, etc. These include disorders such as asthma, diabetes, and cancer. Dear students, let's now discuss genetic counseling. Genetic counseling refers to guidance and support given to patients relevant to their genetic disorders by a healthcare professional called genetic counselor. The National Society for Genetic Counselors defines genetic counseling as the process of helping people understand and adopt through the medical, psychological, and familial implications of genetic contributions to diseases. Genetic counselors are well trained to present complex and difficult to comprehend information about genetic risks, testing, and diagnosis to families and patients. They hold the platform for giving families the discernment to understand the significance of genetic condition at the backdrop of their familial, regional, and cultural contexts. For a genetic counseling session, the counselor interprets the genetic tests designed to diagnose or estimate risk of a disease. For most genetic disorders, genetic counseling is recommended. With the advent of computational biology and next generation sequencing technologies, genetic tests, especially those about DNA sequencing, have become more accessible and relatively affordable as well. The scientific community has also significantly increased the awareness of genetic disorders in populations. Doctors have also started to refer individuals with or without their families to a genetic counselor in their healthcare journey of genetic disorders, treatments, and prevention. A genetic counselor takes people for the session, typically in a hospital setting, to get close to an accurate treatment and understanding. They work in tandem with obstetricians, oncologists, and other specialists. Common diseases that the counselors provide sessions for include the following: prenatal and preconception. Couples susceptible to genetic disorders may also seek prenatal diagnosis to be prepared for the risk or make a better decision regarding the pregnancy depending on the risk. Second, pediatric. Global development delays, hearing impairments. Third, the cancer. Hereditary screening for families. Neurological, individual diagnosis, and family screening. Dear students, we will now understand the role of genetic counseling in the prevention of some diseases. Let's discuss them one by one. First, the breast cancer. Breast cancer is the most common cancer in women worldwide. Genetic screening followed by counseling can help in early detection of breast cancer or tumors. Research has shown that the breast cancer diagnosis can have a hereditary predisposition, so genetic testing and counseling can play a critical part in such an illness. The genetic basis of inherited breast cancer risks had begun to reveal right after the discovery of breast cancer gene 1 or BRCA1 and breast cancer gene 2 or BRCA2 in the 20th century. Even today, these two genes are regarded as the most crucial genes relevant to the genetic predisposition for breast cancer. These genes in healthy conditions are primarily responsible for the error-free DNA repair process and maintenance of genomic stability. Other than BRCA1 and BRCA2, genes like ATM, that is ataxia telangiectasia mutated, or PTEN like phosphatase and tensin homolog, and TP53 like tumor protein 53 are also identified to be associated with breast cancer inheritance. Genetic counseling has become an important tool for providing information and support to families at high risk. So, genetic counseling offers a scope for the breast cancer, that is for early detection of the breast cancer. Dear students, the second one is the role of genetic counseling in neurodegenerative diseases. Some neurodegenerative diseases can be suggested or even predicted by genetic testing. The diagnosis of a neurodegenerative condition has implications not only for the diseased individual but also for the relatives who may be concurrently witnessing the decline of their family member and contemplating their own risk for manifesting similar conditions. Advances in the genetic testing for neurodegenerative conditions have improved the ability to make accurate diagnosis and provide recurrence risk information to family members. It has been noted in many studies that certain neurodegenerative conditions that have a pattern of autosomal dominant inheritance are associated with a 50% recurrence risk. Genetic counseling for diseases like Huntington's disease, early onset autosomal dominant Alzheimer's disease or APP, PSEN1, PSEN2, and amyotrophic lateral sclerosis, that is SOD1, FUS, TARDPP, have started to become more common and significant. Dear students, let's discuss the other condition like cystic fibrosis. Cystic fibrosis is caused by intense dysfunction of the cystic fibrosis transmembrane conductance regulator or CFTR gene and is a life-shortening disorder associated with progressive lung disease. In most cases, genetic counseling for parents of children with CF remains simple, as the main purpose is to provide a clear understanding of the recurrence risk every future pregnancy and how it could be taken care of by prenatal screening. Dear students, let's conclude the lecture. This module has provided a much more detailed understanding of hereditary and genetic counseling. We now know how increasingly important genetic counseling is, especially in routinely being provided to patients with increased risk of carrying genetic disorders like cystic fibrosis, breast cancer, neurodegenerative diseases, and many more. Nowadays, these services are available not only at clinics or hospitals, but also through companies that offer direct-to-consumer genetic testing services. Hope you have got a clear concept about genetic counseling. Good luck.